We are live.
Good evening, all. Welcome to iFocus Online, the 321st episode and the 12th in the Back to Basics module. Today, it is indeed a pleasure to have Dr. Pranesh Balasubramaniam, sir, once again from the Moorfields Eye Hospital, London, to speak to us on Essentials of Ophthalmic Physiology, the Part 2. Dr. Pranesh Balasubramaniam has his MBBS from the Coimbatore Medical College and MS in Ophthalmology from Arvind Eye Hospital in Postgraduate Institute, Madurai. He is currently a clinical teaching fellow at the Moorfields Eye Hospital, London. He was an ex-medical officer at Arvind Eye Hospital, Coimbatore. The primary interests in the field of ophthalmology include ophthalmic education and uveitis. He is a chief educator and founder of the King's Crus, which is an ophthalmology pearl by Dr. Pranesh. Presented in many conferences across India, especially on ophthalmic pedagogy. He is a close-up magician, loves to perform for his friends and family. Welcome, sir. We are ready for your part 2.
Thank you so much, Shubha, for this wonderful introduction. And thanks to Santosh, sir, once again, and his incredible team of fellows for inviting me and for the hospitality. Now, in the previous episode, in the previous part 1, we discussed about a good chunk of physiology covering the tear film, cornea, and the lens, which I think covers 40 to 50% of the chunk of ophthalmic physiology, what you will be asked in examinations. The second half is much more important. We will today cover 2 very vital areas. One's going to be aqueous physiology, and it's going to be the retina physiology. So both are highly dreaded topics, what I call as an FAQ, or which is frequently asked question. And also there is one more way of using the word, the acronym FAQ. It's frequently avoided question also. So it's very commonly asked, but it's not very often addressed, and people are not being taught in the right way, I think. So we'll focus on those two, on those 2 critical areas for today. All right. So my screen is visible. So in the previous episode or previous part, we finished with the accommodation of the lens. That was the where we kind of ended. To start back in this part 2 of ocular physiology, we will deal with the anterior chamber and retina, and we will try to kind of touch upon vitreous to some extent. Okay. Again, this is where I'm going to put all my videos on anat, physio, and more clinical applications on my YouTube channel called Pranesh Balasubramaniam. So you go to this channel and you have this playlist, you click on that, it goes to this, you can find one of the playlists called as a bridge series, where I upload these lectures on anat and physio together and correlating with clinical concepts, hence the name bridge series. So I strongly recommend the readers to watch this video on the angle of anterior chamber, which is part 1. So I've not uploaded part 2 yet, which is what I will be dealing on for you guys. So you can watch the anatomy first, because understanding the anatomy is very crucial to have an idea of how the physiology or how the aqueous outflow works in the first place. So with that basic idea, let's move on to the aqueous humor. So we'll discuss with the physiology, biochemistry, and some important clinical correlates. So aqueous humor, anatomy is something which is very important, and it's not very easy, especially to reproduce in your exams. So that particular video, what I referred, will help you to reproduce the line diagrams in your exams. Physiology is much more straightforward. But what is dreaded, or what is very difficult, is going to be this biochemistry part. So let's deal with the aqueous humor physiology. The one topic which we discuss, which we will focus on, is going to be the aqueous humor dynamics. So what is the dynamics of aqueous humor? We will deal in 2 aspects. One, how the aqueous is going to get formed and how it's going to get secreted. That's going to be first part of this. The second part is going to be how the aqueous is going to get out and how it's going to enter into the exiting pathway of the trabecular meshwork. So we'll deal with the 2 types of flows, which you might be aware of. So we'll deal with the formation and secretion of the aqueous humor now. As you all know, there is going to be these 3 routes of exit. Let me just connect once again. There is going to be these 3 routes. Okay. We all know that aqueous is going to be formed from your ciliary processes, and they're going to come out through this. They're going to enter the pupil and they're going to drain into the trabecular meshwork. Basically, there is going to be 3 routes of exit. Okay. In other words, how the aqueous is going to be formed, how they are going to travel out. That's going to be 3 routes. First is going to be this route, where the aqueous is going to come out of the blood vessel and is going to enter into the stroma. So the uptake of solute in water from the plasma into the stroma is going to be number 1. Number 2 is going to be when this aqueous is going to form and is going to enter into the pigmented epithelium. In this picture, you can see this red color is going to be this pigmented epithelium, whereas the blue is going to be the non-pigmented epithelium. So first will be this coming out. The second is going to be how it's going to get transferred from this pigmented epithelium to the non-pigmented epithelium. The third is going to be how this non-pigmented epithelium will release the aqueous into the posterior chamber. So there are going to be 3 routes or 3 stations it takes. To put it more simply, like I said, it's going to be the blood vessels, the capillaries there, and they're going to give out the aqueous. So what is aqueous? They're nothing but an ultrafiltrate of plasma. So this aqueous is going to come out. They're going to enter into the pigmented epithelium. So from the pigmented epithelium, they're going to enter into the non-pigmented epithelium. From the non-pigmented epithelium, they're going to exit into the posterior chamber. So far it's clear? Okay. And these gap junctions, which are here, play a very crucial role in maintaining integrity, and they will not let anything else to happen to the aqueous, which is very important, which we will discuss in the blood aqueous barrier. Okay. Now, there are 3 modes of transport actually. So now we know how it's going to come out. We saw the 3 ways how it's going to exit out. Now, we will look at the various mechanisms which help in this exiting out of the aqueous from the ciliary processes into the posterior chamber. First going to be this ultrafiltration, which means it goes along the pressure gradient. The second is going to be the use of active pump mechanisms. The third is going to be the passive diffusion. So please understand, this passive diffusion means the passive diffusion of water. So we have these 3 mechanisms by which the aqueous is going to come out into the posterior chamber. Now, let's see how these mechanisms and where they act. This ultrafiltration is going to be happening at this area where the aqueous is going to exit out from the stroma, from the blood vessel into the stroma, from stroma into the pigmented epithelium. This is where the ultrafiltration comes into play. Whereas the active transport or the use of the active pumps happen in 2 areas. One is going to be at this junction between the pigmented and the non-pigmented epithelium, and second is going to be from the non-pigmented epithelium to outside to the posterior chamber. So this is where the active pumps play a role. The third is going to be the passive diffusion of water goes from here to the posterior chamber. Now, this might look like a very dreaded picture, but I think so far it's very clear that you have these 3 ways of which aqueous is going to goes out. Now, before I explain this, I just want to give you a minute to just see this and appreciate the complexity of it. You will understand that it looks quite complex, but in fact, it's very simple. So remember the principles what I discussed in the previous class. I said whenever sodium goes out, water goes out. Whenever sodium gets in, water also gets in. So sodium and water are like very close friends. They're like in a relationship. So both of them always travel together. Okay. So sodium should go out so that water should go out. That's the first thing you should understand, because aqueous is nothing but water. So you need water to be transported outside. So here's going to be the water which goes out from your ciliary process into the posterior chamber. So water should go out by these aquaporin channels. When does water go out? Water will go out only when the sodium is going to go out of the ciliary process, from the ciliary epithelium. Only if it gets out, water also will go out. So for clear? Now, when the sodium goes out by this pump, by the sodium potassium ATPase pump, which is located at this border between the posterior chamber and this pigmented epithelium. Clear? The non-pigmented epithelium. So at this junction, there you have your sodium potassium ATPase pump. Now, this sodium potassium ATPase pump is activated by a beta adrenergic receptor here. Okay, so this beta adrenergic receptor is going to facilitate sodium to go out so that water also gets out. Now, when this is blocked, when you have a beta blocker coming here and blocking this particular receptor, what happens? The sodium potassium ATPase pump will not be activated. So sodium will not go out. Water does not go out. So it's reduction of aqueous secretion. It's a clinical correlation, which we'll discuss again in a minute. Now, look at the other equation here. For sodium to go out, sodium should come out of the stroma into this epithelium part. That's the crucial thing. So for sodium, once it comes out of this blood vessels into the stroma to enter the epithelium, it has to be exchanged by a sodium hydrogen like an exchanger. So it's going to be an exchange pump, sodium hydrogen exchange pump axia, so that the H plus should come inside and sodium should go outside. Only when sodium goes outside, it can go all the way, it can get out, and you have sodium going out, water going out, aqueous forming. Now, how this H plus is being formed? This H plus ions are being formed by this reaction which happens here. The water combines with the carbon dioxide, okay, to form HCO3, and it gives this H plus ions. Now, this is an important reaction which is mediated by an enzyme called as carbonic anhydrase. So what happens here? It's because of the carbonic anhydrase that H plus is being secreted, and only because H plus is there, sodium gets exchanged, sodium goes out, water goes out. So what happens when you have a carbonic anhydrase inhibitor? You have a carbonic anhydrase inhibitor. This is going to get inhibited, so H plus is not going to form. Therefore, your sodium is not going to be exchanged. Sodium is not getting out, so no water getting out, reduced aqueous secretion. Another important clinical correlate or applied pharmacology aspect of it. So this is going to be the brief basic mechanism here. So I want you guys to reproduce this picture in your examination. The only thing what you can do is you can put the same pump here as well. You can put the same reaction here as well. So that way, so now we have described how the aqueous is going to get secreted out of your epithelium and how it's getting into the anterior chamber. I hope things are very clear. Okay. So to review again, it's going to be from the stroma, it's from the blood vessels entering the stroma, from there to the pigmented epithelium, to the non-pigmented epithelium, and from there exiting to the posterior chamber. So sodium is going to get out, water is also going to get out by this passive diffusion. Therefore, you have aqueous formation. So now you know how aqueous is formed and how is it getting secreted. So like I said, there is going to be three important areas, two of which we discussed. One is going to be the beta blocker, which blocks the sodium potassium ATPase transport. So no sodium, no water, no aqueous. Carbonic anhydrase inhibitor, which again, the carbonic anhydrase is going to be inhibited, as a result of which the H plus is not going to get exchanged with sodium. So no sodium goes out, no water goes out, reduced aqueous. The third is going to be this alpha agonist. This alpha agonist is going to cause, normally the alpha receptors are going to be there. This alpha agonist is going to cause vasoconstriction. So some books says it causes vasoconstriction. Some says it reduces the ciliary body blood flow. So no matter what the mechanism is, there is going to be reduced secretion of aqueous, okay, from the blood vessel. That's where the alpha agonist comes into play. So you have this A, B, and C, which are three important drugs which are used to reduce the production of aqueous humor, three important drugs in your treatment of glaucoma. Okay, I hope I've covered the pharmacology part a bit so that now you understand the importance of this. So in exams, if someone asks you about the formation of aqueous or the aqueous outflow, you have to talk about this aspect as we have talked about how aqueous is going to get formed and secreted, and you will get brownie points. You will really get exceptional marks if you make sure that you put this in your examination. So always combine the clinical aspects with your physiology as an applied physiology. So now we finished about the aqueous formation and secretion. Now we talk about the aqueous humor outflow. Now, this is something which you guys already know about in a lot more detail than what I'm going to teach you now. But again, this aqueous outflow is frequently asked exam question. So please remember this. You can see that there are two ways by which the aqueous is going to get out of the system, get out of the eye. And one of the ways is going to getting out through the trabecular meshwork. This is going to be the conventional trabecular outflow. On the contrary, you have something called uveoscleral outflow, which is a minor thing, where the aqueous is going to pass through the ciliary body band. Okay. Now here you can see this is going to be the bigger blue line is going to traverse the journey of the trabecular outflow. So from the ciliary processes, it goes through the pupil and enter the anterior chamber. The anterior chamber, the aqueous is going to get into the trabecular meshwork, Schlem's canal, exit out. On the contrary, you have this uveoscleral outflow where you have this small blue line here. You can see a part of the aqueous once in the anterior chamber. It's going to go through the ciliary body band, then into the suprachoroidal space, and then exiting out. So you have two important outflows there. This is a chart which is given in Dr. Kurana's Anatomy Physiology book, which will be the standard book which you guys will be following. So this is going to be a very important, very comprehensive table there. The aqueous humor have the trabecular outflow, uveoscleral outflow, trabecular meshwork, Schlem's canal, the collector's channel, episcleral veins, superior ophthalmic vein, and the cavernous sinus. This is basically an anatomy part, but I'm just bringing this into the physiology as well. Here, the aqueous is going to have this uveoscleral outflow where it goes the root of the iris and ciliary muscle or the ciliary body band, suprachoroidal space, exiting through intact sclera or penetrating nerves or vessels, orbital veins, superior ophthalmic vein, and cavernous sinus. So the end point is same. It goes to superior ophthalmic vein and the cavernous sinus, but you have two different routes to reach that cavernous sinus. This is an important table, the important flow chart. Now, to compare and contrast both, trabecular outflow is going to be more of a pressure sensitive or a pressure dependent. So it's more dependent on the intraocular pressure, whereas uveoscleral is going to be a pressure insensitive or pressure independent. It's conventional, which means the majority of the outflow, almost 80 to 90%, is by the trabecular outflow, whereas the unconventional, the 10 to 20%, is by the uveoscleral outflow. The trabecular outflow is going to be increased by pilocarpine, whereas uveoscleral outflow is going to be increased by your prostaglandin analogs. So just remember these very primitive and basic differences between these two outflows. Now, what are the clinical implications of this? Now you can see any block in this. For example, when this exiting out of aqueous into the superior ophthalmic, when this is not going to happen. In other words, what happens when here there is a problem there in this draining? There's going to be a rise in the episcleral venous pressure. A rise in episcleral venous pressure is an important clinical concept to understand, which often presents as the corkscrew vessels on the bulbar conjunctiva. And if you can recollect, where you will have this rise in EVP or episcleral venous pressure, it's going to happen in your thyroid disease, your cavernous sinus thrombosis, in your keratico-cavernous fistula. These are some of the many conditions which can cause a rise in the episcleral venous pressure. So you can expect this corkscrew vessels in these patients. This is an important concept. Again, Goldman equation is very often expected in your exams, and they may ask you in your viva also. So what is this? It's going to be a very basic idea. The rise in episcleral venous pressure. So what this is going to lead into? The normal value of episcleral venous pressure is going to be 6 to 9, contrary to the normal value of intraocular pressure, which is 10 to 20. Now, for every 1 mm rise in the EVP, there's going to be 1 mm rise in your IOP as well. This is just basic data to understand. So EVP is always less than the IOP, but both are directly proportional. That is the concept. Now, this is the equation. So please focus on this. IOP is equal to rate of aqueous formation divided by the aqueous outflow facility plus EVP. It's nothing but more IOP will happen when there's going to be more of aqueous formation. The rate is going to be more. And more IOP will happen when you have a less outflow facility. So both are like, this is directly proportional, this is inversely proportional. This is going to be the equation. You just put a plus EVP, done. This is going to be the Goldman's equation. Okay. Now this is just some numbers and some kind of alphabets they have assigned to each. So you can just remember this. concept is when you have a rise in EVP, there's going to be a rise in IOP. So more IOP, more EVP. The vice versa also happens. So more epically venous pressure is going to increase your intraocular pressure as well. That's why you can even have a high IOP in thyroid eye disease. More IOP, like I discussed, more IOP is going to be more of rate. More IOP means less of outflow. Okay, this is going to be the basic Goldman's equation. So with this, we finished an important concept called as the formation and secretion of aqueous, which is my primary focus, because the outflow is something which you can easily read. But what will be more difficult will be how it is formed, how it is secreted. That biochemical reaction, that is going to be the difficult part which I addressed. Hopefully that's clear. Now let's move on to the biochemistry of aqueous. Like I mentioned in the previous lecture, part 1, this is a very important diagram, which I think you can always have this in mind. There's always going to be less sodium and less water in your cornea as well as in your lens. You should always have relative dehydration in both these organelles, along with a relative hyponatremia, or hyponatremia means reduced salt in the blood, but here reduced salt in your lens and cornea. So yeah, you need both should be less. On the contrary, there'll be more of sodium and there will be more of water in your aqueous, which is again is clearly depicted from this table. So you can see the aqueous humor will have more of sodium, more of chloride, more of ascorbate when compared to plasma, and they will have less of glucose and less of proteins. Clinically very important. Now, sodium and chloride are going to be the salt. Like I said, sodium is going to be more in your aqueous, which you now know, because you have the sodium kind of getting out, and we saw that when sodium comes out, your water comes out. In fact, sodium and water is going to be more important constituents of the aqueous humor composition. But this ascorbate should be more, because ascorbate is going to be like a free radical scavenger, like a glutathione. It is going to fight against lots of evils we just discussed in a minute. Apart from that, you need to have less of glucose and you need to have less of proteins as well. So less of glucose means what? You should not have. I know that glucose should not be there anywhere. Glucose should not be there more in your lens as well, and should not be there more in your aqueous as well, because more of glucose in the aqueous again can lead on to that glucose getting into the lens, causing a cataract. And proteins, you should never ever have proteins. And you all know that when you have proteins in your aqueous, that's where the aqueous becomes from a clear liquid into a turbid liquid. We'll discuss about this in the blood aqueous barrier. There are some more concepts to understand here. When it comes to aqueous humor, there's going to be these 3 G's which are important. It's going to be glucose, glutathione, and these growth factors. Like I said, glucose should be reduced. Glutathione and ascorbic acid are going to be these protective mechanisms, and you even have these growth factors like your TGF beta, transforming growth factor beta, insulin-like growth factor 1, and your VEGF, vascular endothelial growth factors. So in fact, this growth factors in the aqueous means that the ciliary epithelium might have a neuroendocrine property. This is like a recent update. Okay, and not just going to be these, even the importance of oxygen and carbon dioxide are quite significant here. Now, what happens when you have more of glucose? When you have more of glucose, like I said, there's going to be a formation of sorbitol into the lens, and more of sorbitol, water is getting drawn inside, loss of transparency, metabolic cataract, just to revise the previous lens, the cataractogenesis in high glucose. Now, speaking of blood ocular barrier. Now, this blood ocular barrier is going to be of 2 areas, the blood aqueous barrier and blood retinal barrier. But recently they have given this blood vitreous barrier also. But in exams, if someone asks what a blood ocular barrier, you can just mention these 2. This is just something extra for you to know. The blood aqueous barrier is basically formed by 3 important components. One is going to be the non-pigmented epithelium of the ciliary body. Remember this non-pigmented epithelium here? The non-pigmented epithelium of the ciliary body, this blue color, is going to be the first and important component of the blood aqueous barrier. The second component is going to be this presence of iris vessels. You can see these iris vessels here, which again forms this blood aqueous barrier. And the third component is going to be the inner wall of the Schlem's canal, which you will not remember. What you will remember is just this. This is what we all normally think, but you need to add these 2 also. One's going to be the iris vessels, second is going to be the inner wall of the Schlem's canal. So the tight junctions in these areas, the tight junctions of the non-pigmented epithelium, iris vessels, and inner wall of stem skin, all they're going to be protective. They're going to prevent the leakage of blood constituents into your aqueous, therefore maintaining clarity of the aqueous humor. Now, I think I might as well cover the blood retinal barrier here also, because I will not be talking about this when we deal with the retinal physiology. So blood retinal barrier is again have two components, the inner blood retinal barrier and the outer blood retinal barrier. The inner barrier is going to be formed with the inner retina. So what we have in the inner retina, you have the retinal capillaries. So the tight junctions of retinal capillaries, okay, they're going to promote this inner BRB. Whereas the tight junctions of the RPE, retinal pigmentary epithelium, you have this outer BRB. So for clear? So any breach in the blood aqueous barrier, you will be having uveitis. Any breach in the blood retinal barrier, you will have exudations, you have hemorrhages, you have heart exudates, all because of breach of this blood retinal barrier. What is blood vitreous barrier? Someone asks you what is blood vitreous barrier. Just mention these two. It's a blood aqueous barrier, blood retinal barrier. Along with that, you have the cortical vitreous or the peripheral part of the vitreous is going to be another component of the blood vitreous barrier, and the basal lamina of the vitreoretinal junction. Just know these concepts. So please remember that you have something else called a blood vitreous barrier. In an exam, if you can add this, you may get more marks or brownie points. Right. So we discussed about the blood aqueous barrier, and we saw the importance of this blood aqueous barrier where to maintain the clarity of the aqueous, to maintain this protein free. Now, there are multiple causes why you have a breakdown of the blood aqueous barrier. More common is going to be the trauma, drugs. So trauma can be either mechanical or surgical trauma, which means post-cataract also can experience uveitis. There is a breach of the blood aqueous barriers there. Use of 2-Ps, the prostaglandin analog and pilocarpinol, can cause an increased AC reaction, can cause uveitis. So these two drugs, ischemia in a condition such as ocular ischemic syndrome, though it is essentially retinal, you can also have AC reaction, ocular ischemic syndrome. Anteruveitis. So of various pathologies, even trauma can cause anteruveitis. So what here mean is the autoimmune or infective causes of the anteruveitis. So all these things can break the blood aqueous barrier. And how they manifest as AC cells and AC flare. Cells are nothing but the white blood cells, the inflammatory cells in the aqueous. Flare is nothing but the protein leak. When you have more of flare, it's to flare 4+, that's what you call as a fibrinous membrane, or what you call as a plastic iridocyclitis, a severe form of acute anterior uveitis. Just some clinical correlates. So in exams, when someone asks you this question on blood retinal, the blood aqueous barrier, you start from the blood ocular barrier. Then you take a travel down. This way, the examiners will understand that you know much more and you have a better conceptual idea where to put where. So quickly moving on to vitreous. So I'll just quickly focus on vitreous just for a few minutes, because what follows is going to be much more important. So this vitreous considers like a small short break. I thought vitreous will never be asked in your exams, but to my surprise, vitreous is one of the frequently asked exam questions. For MS, especially for MS and DO, we don't have DO these days, but for masters and for government institutes, vitreous is going to be very important, high yielding topic. I think it's the same for DNB as well. Okay. I'm bringing back this table once again to show that, to compare how vitreous is going to score against aqueous. So both are going to be important structures which maintain the integrity of the eyeball. The sodium is going to be less. The chloride is going to be less when compared to your aqueous. But vitreous has more of ascorbate. Okay, that's an important thing to have in mind. Which means vitreous needs more of ascorbate to fight against the free radicals there. So you need more of ascorbate there. Like always, you need to have less glucose in your aqueous, in your vitreous as well. Okay. So this is a very easy picture where you can see that the vitreous is going to give glucose to your retina and to your lens. That's why vitreous is having less glucose, because retina is a quite metabolically active structure. That's why it keeps on constantly giving glucose more to the retina than to the lens. But vitreous is less than depleted in glucose because it has a constant supply to the retina. And like I said, aqueous is going to supply glucose to your cornea as well as to your lens as well. Now, this is a picture to show that how clear the vitreous gel is. It's so transparent and it is so thick and viscous. Just to refresh the anatomy, vitreous, you have two components. You have the central core vitreous, you have a peripheral cortical vitreous. Now in the vitreous, basically it's going to have collagen. So collagen is going to be a common composition in, be it in vitreous or cornea. You can see the collagen fibers play a huge role there. The collagen fibers are going to be 2, 9, 5/10. So there are going to be 3 important collagen fibrils. So these collagen fibrils are suspended in the cement substance called as the glycosaminoglycans. And this glycosaminoglycans can be of 2 types in vitreous. One is going to be chondroitin sulfate. Second is going to be this hyaluronic acid molecules, which are hydrated. This hydrated hyaluronon or hyaluronic acid is going to maintain the viscosity of the vitreous, maintains the structural integrity, keeps the vitreous like a gel. So this is an important element in vitreous, which keeps vitreous as vitreous. So we just deal one by one in brief. So collagen fibrils, you have, this is going to be structure of the collagen in vitreous. So please draw this diagram in your exams as well. You have like an insulation, which is the type 9 is going to cover the type 2. The type 2 is going to be the most common type of collagen seen in vitreous. And inside, it's like a wire, it's like insulation wire. You have like outer 9, middle is going to be the number 2, and innermost going to be the type 5/11. So for clear, so of this type is going to be most common. Why I'm bringing this into picture is, in exams, if you're just going to put this, it's good. But what's even better, you're going to give an applied aspect of this. So what if this gets mutated? So any mutations of collagen 2 is going to cause a Stickler syndrome, which is characterized by this optically empty vitreous with high myopia and recurrent and high rates of retinal detachment. So Stickler syndrome has a very direct correlation with mutation of collagen type 2 in vitreous. Coming on to glycosaminoglycans, like I mentioned, it's going to be this hydrated. So the complex between the sodium, water, hyaluronan is going to maintain this viscosity of your of the vitreous. Hyalocite is going to be the most predominant type of vitreous cell. And like your ACAD or anterior chamber associated immune deviation, you have this VCAD, where it's a vitreous cavity associated immune deviation. So both have to be immune privileged. That's why when you have a breach of the blood vitreous barrier, you have vitritis. Okay. Now, what is the importance of these hyalurocytes? Another important clinical correlation is that these hyalurocytes are implicated in the formation of this epiretinal membrane on the macula. Okay. Now, coming to an important area, there is the changes in vitreous. What happens to vitreous in physiological, in pathological, in case of iatrogenic? In physiological change of vitreous, as age progresses, it undergoes a change called as PVD. PVD, or what you call as a posterior vitreous detachment. Pathologic change is going to be of 2 types. In myopia and case of PDR, or proliferative diabetic retinopathy. When you have a myopia, there's a premature vitreous detachment. There's going to be this premature aspect of it, which means in myopic patients, there's going to be a faster liquefaction, faster changes in vitreous. So you will see in lots of young myopic patients, there will be PVD. PDR, just want to give just a quick thing. There'll be rise in VEGF, which you all know. But what is more important, there are going to be these 2 important substances, 2 important chemicals in vitreous, thrombospondin and PEDF, pigment epithelial. Sorry, I forgot the name. So PEDF. So these 2 are going to inhibit the VEGF increase. So both are going to be having in the normal vitreous. Okay. What is iatrogenic means? Post PPV, post vitrectomy, we'll see what changes will happen. So one by one, we'll take it. Now, what happens in your PVD? So PVD is going to be 2 important. It's going to be synchisis and syneresis. Synchisis means there's liquefaction. Syneresis means it's going to be condensation. Okay. What happens in this liquefaction? There's going to be a very important thing happening here. You can see that once the vitreous is going to get liquefied. Why it becomes liquefied? Because the collagen fibrils inside the vitreous becomes fragmented, therefore it forms this liquefied vitreous. Now, when you have a small hole in the posterior cortical vitreous, which is the peripheral part, now this vitreous can kind of becomes liquefied or becomes kind of this synchisis. It goes down and is going to separate, it's going to detach the vitreous attachment from the retina, therefore causing this PVD. Okay, so this is going to be the aging changes in the vitreous causing PVD. All right. So we finished the physiological aspect of it. We finished about the pathological aspect of it. Okay. I think this is pigment epithelium derived factor. Okay. Now, what happens post vitrectomy? You would have seen in patients who had underwent vitrectomy or passed a vitrectomy in phakic. When you have a clear lens, you do a vitrectomy, you can see after some time they can develop a cataract. Why that happens? When you clear off the vitreous, you're going to create more empty space, and that facilitates more of oxygen there. We discussed in the previous class that oxygen is going to be an important villain. So we don't want much of oxygen there. So you have this oxygen species or reactive oxygen species coming out of this oxygen, and there they can cause cataract. And not just that, they can even cause open angle glaucoma as well. As you can see in this table, which I brought in the previous lecture as well, you always please understand these three important evils and three important saviors of the oxidative damage. Hope I've covered quickly vitreous. Vitreous is a high-yielding topic, but this retina is going to be even more important topic, primarily because you have different books explaining in different ways. So what I want to give is I want to give a more of like a structured, more of like a proper way of understanding this. I will deal with two important areas. It's going to be the biochemistry. Second is going to be the physiology. In biochemistry, we will deal with the photochemistry of vision. In physiology, we'll deal with the phototransduction pathway. So both of these two topics are going to be frequently asked exam questions. So we'll take it one by one. Now, this diagram, this particular flow chart is something which I made as the last slide of this talk. I realized then that this should be one of the first slides because understanding helps you to put everything in the right perspective. Okay, so just have a minute and just have a look at this image. So let me just talk you through this. Vitamin A is going to be one of the important vitamins which we need for the formation of your retinal pigments, which you all know. Be rods and cones. So we all know that how the importance of having carrots for a good sight, which is actually a myth. But this vitamin A is going to be obtained from the diet. Okay, from the diet, vitamin A comes, and from the body it goes to the blood. So from the blood, you have the vitamin A, which is like the retinol. There's going to be the form in which it is going to be present or existing. From the blood, it goes to the retinal pigmentary epithelium. So in RPE, there's going to be retinol, which becomes this 11-cis-retinal. 11-cis-retinal. So this alcohol becomes an aldehyde. This cis indicates the position of the atoms in the particular molecule, which is not needed for us. But just remember, 11-cis-retinal. Now what happens? This 11-cis-retinal will go to the rods or cones. It goes to the photoreceptors, especially the outer segments. I will discuss everything in detail, but I'm just going to give a brief overview initially so that it makes much more understanding for you. Now, this is going to go into the outer segment of rods. In outer segment of rods, which means the outer part of the rods or cones, you have these disc lamella. Okay. So what happens? This 11-cis-retinal gets into the outer segment of rods. It's going to get attached to this particular protein called as a rhodopsin, which is going to be like a snake or like a wire wound around the discs. You have many rhodopsin in each disc. So I'm just giving a representation of each. So this 11-cis-retinal is going to gets attached to this rhodopsin. Now what happens when the light strikes? When the light strikes or in daytime, this 11-cis-retinal is going to become all trans-retinal. And not just that, what is more important is why it should happen. Because rhodopsin gets activated on light getting struck, rhodopsin is going to induce lots of changes, what we call as a phototransduction pathway, which we'll discuss again in a minute. Now, this 11-cis-retinal, which has formed, is going to become all trans-retinal. This all trans-retinal is going to become all trans-retinol. So when light occurs, you see activation of rhodopsin, and you also see this 11-cis-retinal becoming all trans-retinal. Now, this all trans-retinal is going to become a retinol, and this is the thing which kind of travels back to the RPE. Please understand, travels back to the RPE, And this is going to get converted back into a Levensis retinol, which becomes Levensis retinal. I hope I've made this clear. Now, this is going to be the entire summary. So this process by which this Levensis retinol becomes Levensis retinal, which means we have regenerated vitamin A. We have regenerated this Levensis retinal. This is called as the Wall's visual cycle. Okay, this is a Wall's visual cycle. So you can see that this retinol has become retinal. And not just that, this retinal is also going to be formed from the diet resources of your vitamin A. So you can have a constant regeneration of vitamin A, both from our diet and also by itself. It's going to regenerate vitamin A in the form of this Levensis retinal. So we will discuss first this phototransduction concept, and then we will end with this. So there is lots of details, lots of intricacies with this phototransduction, and there's going to be more of concepts discussed in this part also. We'll take one by one. So first we'll start with this. We'll deal with the vitamin A sources, how they get absorbed, how they get stored, how the vitamin is going to get transported from liver to the eye, and how vitamin is getting utilized. So in all, in a nutshell. So the diet vitamin A, you have either animal or plant resources. This animal, you have meat or liver, milk. Milk and the meat, 2 M's, actually give the actual vitamin A, which is the retinol. So meat is a rich source of retinol or vitamin A. Plant gives these precursors, namely the alpha, beta, and the gamma carotenes. Now, what happens to this retinol? This retinol is going to go into the stomach. Okay, it's going to get absorbed from the intestine. From there, through the blood, it gets stored in the liver. So liver is going to be the most concentrated source of vitamin A. So there's going to be storage of vitamin A in the form of retinol. And what happens? This retinol is going to come out of the blood. In the blood, they're going to bind to this protein called as retinol binding protein. Retinol binding protein. So in the blood, you have the retinol, which is a dietary vitamin A, is going to combine retinol binding protein. Now what happens? This blood is going to give the retinol to the retinal pigmentary epithelium. So after liver, the most concentrated area where you have more vitamin A is going to be a retinal pigmentary epithelium, or RPE. So RBP, this binding, it's like a taxi driver. So it's going to kind of drops this retinol into the RPE. Now what happens? Like I said, this retinol is going to become Levensis retinal. Levensis retinal. So this all is going to be an alcohol, is going to convert it into an aldehyde, which is this Levensis retinal. This Levensis retinal is going to come out of the RPE. It's going to get into the rods and cones, to be more specific, the outer segment of rods and cones, even to be more specific, into the disc lamella. They're going to combine with this opsin, which is a protein called opsin. Okay, it's going to form this rhodopsin. Some textbooks will call rhodopsin by itself. They will consider rhodopsin and Levensis retinal as separate entities, but actually Levensis retinal is going to combine with this protein. This protein is called as opsin. Here in rods, it is called as scotopsin. Scotopsin. So this is going to combine with scotopsin to form this rhodopsin. So rhodopsin is nothing but a combination of Levensis retinal with scotopsin, also called as visual pigment. But in our explanation, we will never use the term scotopsin. We will always talk about rhodopsin. Rhodopsin and this Levensis retinal. I think so far it's very clear. Okay. So this Levensis retinal is going to combine with this opsin in the outer segment of rods and cones to form rhodopsin, which is also called visual pigment or visual purple. Now, to summarize the same, from the diet, it goes to the stomach, intestines, liver, from liver to the blood. Blood, it binds with the retinol binding protein. From there to the RPE. From RPE, the retinol becomes Levensis retinal that gets transferred to the outer segment of rods and cones, combines with the opsin to form the rhodopsin. Hope everything is clear as of now. Now, just a brief thing about the photoreceptors. So you all know that photoreceptors, you have rods and cones. I'm not going to give the anatomy part of it, but just to have the relevant of anatomy concepts here. You have this inner segment, you have this outer segment. For both photoreceptors, rods and cones, you have this inner segment, outer segment. You see in this outer segment, you have this multiple discs called as the lamellar discs. You have rod discs, you have cone discs as well. Now, What happens on the disc membrane? Now, for all study purposes, we will consider rods. We won't consider cones. So this is one question which often I've asked myself, why people always talk about rods? What about cones? Despite the fact that cones are going to be more important for actual vision. You're very right. Cone is going to be more important than rod, because rod is what subserves the peripheral, the night dim vision, whereas cone is going to subserve. They're going to be playing an important role in your color, contrast, curvature vision as well. For clear vision, you need cones, very importantly. But rods are more studied than cones. That's one reason. And rods are more in number, and the rods are much more sensitive actually than cones. So for all practical purposes, we will deal with rods, but understand the mechanism of phototransduction is very much the same as the rods. So rods and cones have very similar, not similar, will have they have almost the same way of the phototransduction or how things happening. So the mechanism is going to be same. So in exams, if someone asks you cones or rods, you can put the same answer again. So far it's clear. So outer segment is going to will be having this lamellar discs. Now you take one lamellar disc. In one lamellar disc, you will have multiple this transmembranous serpentine-like protein called as rhodopsin. You will have millions of rhodopsin on each disc membrane. Okay, so just have this in mind. So this is a disc membrane. You have this rhodopsin, which kind of covers this disc membrane. You have like 7 coils. So rhodopsin, like I said, contains a scotopsin with 11 cis retinal. So far it's clear. Now, what happens when light strikes? What happens in daytime? You have 2 important reactions to consider. The first reaction is this 11 cis retinal, okay, which is already there here. This 11 cis retinal, the aldehyde form, is going to get isomerized into all trans retinal. It's nothing but when light happens, the configuration of the atoms inside that molecule kind of changes, kind of flips around to become all trans. So 11 cis becomes all trans retinal. This is an important equation. It's the first equation which happens. The second equation is this rhodopsin is going to get activated into an activated form of rhodopsin. This activated form of rhodopsin is also called as a meta-rhodopsin 2, which means when light comes, rhodopsin undergoes lots of intermediate reactions, and finally it ends with this meta-rhodopsin 2, nothing but the activated rhodopsin or R+. Now look at this diagram here. Now rhodopsin becomes this bartho-rhodopsin. So rhodopsin becomes this bartho-rhodopsin, bartho-rhodopsin, that becomes a lumi-rhodopsin, becomes meta-rhodopsin 1, meta-rhodopsin 2. Just these in intermediates. Just know this, please. Meta-rhodopsin 2 is going to be the activated form of rhodopsin. Now, this activated form of rhodopsin is going to stimulate a protein called as transducin. Very important. It's going to stimulate transducin. This transducin is going to again stimulate this enzyme called as phosphodiesterase. Phosphodiesterase, what it does, it is going to be involved in the conversion of cGMP, cyclic guanosine monophosphate, into GMP. So cGMP becomes GMP, okay, with the help of phosphodiesterase, which is more activated when you have activation of rhodopsin. So what happens when there's more conversion of cGMP into GMP? The cGMP will be reduced. cGMP is reduced. There's one more important thing here. The cGMP, how it is formed in the first place, cGMP is converted to GMP by phosphodiesterase, but this cGMP is going to be formed from this guanosine triphosphate with the help of this enzyme, called as guanylyl cyclase. Guanylyl cyclase. GUA. Okay. Now I've drawn the same here. This is going to be the actual thing just happening in the membrane, on the disc membrane. This R is going to become R plus, or this meta-rhodopsin 2. It's going to activate this transducin molecule. This is going to stimulate this PDE, or phosphodiesterase. It's going to cause cGMP to GMP. Therefore, it's going to be reduced cGMP. So all these things happen when you have light striking the rhodopsin. Clear? Now, what is the work of this cGMP? The cGMP is like a night watchman. Remember, I always remember like cGMP is going to be like a night watchman. So what he does, he's going to open up the gates for sodium at the dark. So at night, CGMP opens up the sodium channels at the dark. Now we said what happens when you have light hitting, CGMP is going to get reduced courtesy the activation of rhodopsin. It's going to be reduced CGMP. So what happens if reduce CGMP? It's going to close the sodium channels. Now, there are some basic concepts to understand. You have outer segment, you have inner segment of the rod or cone. This inner segment, normally you'll have more of negativity inside. Now, what happens normally, sodium gets out of this inner segment. It tries to go into the outer segment through the sodium channels. Now, if the sodium always comes out, not just sodium, even calcium also comes out. So sodium comes out into this extracellular fluid. Okay. Now, it cannot go back into the outer segment. Why? Because sodium channels are closed. Now, sodium channels are closed, as a result of which sodium is going to get retained outside. So, it's going to be more of sodium outside and less of sodium inside or the inside of this rod or cone. Okay. Now, what happens here? There's more of positivity outside and more negativity inside. So, there is a polarity, poles, opposites is going to happen. Now we say that rod is becoming hyperpolarized. Rod has become hyperpolarized. Please remember that. On light, hyperpolarized. Light, high. Okay. On light becomes hyperpolarized. What happens if rod is getting hyperpolarized? When rod is getting hyperpolarized, it is going to inhibit glutamate secretion. Please understand. This is a very important concept to have it in mind. The rod hyperpolarization, okay, is going to inhibit stimulation or inhibit release of glutamate from the synaptic terminal. If glutamate is not released, okay, there is going to be depolarization of bipolar cells. You will remember that during our first physiology that when there is going to be a depolarization, which means there is going to be current. There is an excitatory postsynaptic potential. There's going to be current is getting transferred. You have a depolarization. You have a hyperpolarization, you will not have current. This is what you should remember from your first year physiology. This is quite unique in photoreceptors. Probably the only area in the body where a hyperpolarization is inducing depolarization. A hyperpolarization of the rods is causing depolarization of bipolar cells. The depolarization of bipolar cells is going to cause depolarization of your ganglion cells. Depolarization of ganglion cells is going to cause electric impulses into the retinal nerve fiber layer, to the optic nerve. So you see things in light. Now the question arises, glutamate is going to be an excitatory neurotransmitter, but why and how in the world does glutamate or not releasing glutamate can cause this? Glutamate should be released, right? Only if glutamate should be released, it should excite this, causing depolarization. That's what our common sense would think. But here the ulta happens, the opposite thing happens. Why? This is a question which everyone will have, but the answer for this is kind of difficult or kind of a confusing thing, but I'll make it as simple as possible. There are two types of bipolar cells. You have like off bipolar cells, you have on bipolar cells. This off bipolar cell is nothing but the glutamate is going to excite this off bipolar cell. Off means what? It will not activate. It's an off. It's like a sleepy thing. Glutamate is going to inhibit the on bipolar cell. So here in our case, what happens? Glutamate is not happening. So there is no glutamate. What happens? You will have less of off, but more of on. Because glutamate is not there. So when glutamate is not there, okay, what happens now? You'll have more of on bipolar cells. So there's going to be one of the on bipolar cells. So when the on bipolar cells are switched on, stimulated, this is going to have depolarization. So please remember, in light, a rod is hyperpolarized. In light, BPC is depolarized. I hope I've made things clear for you. Now, like I said, when your BPC, your bipolar cell gets depolarized, your ganglion cell becomes depolarized. There's going to be transmission of the electric impulses here. Okay. So to revise, I said the light is going to hit, rhodopsin becomes active, reduced GMP, the sodium channels close. Sodium channels close, which means there's more sodium outside, there's negativity inside, hyperpolarization, not release of glutamate, which leads to depolarization of BPC, depolarization, impulses travel. Oh, so for clear? So this is where a photon or light, photon or the light energy is getting converted into an electrical impulse, or conversion of light energy into an electrical energy. This is called as a phototransduction. Phototransduction. As simple as that. In its simplest form, you see a light is hitting me, but light is becoming like an electric current, travels to the brain via the neuronal circuit so that I'm able to see what I'm seeing. Now, what happens when darkness comes? What happens when darkness prevails? Everything is ulta, just the opposite. So now what happens? This is what happens when light occurs. So when light comes, we saw that we have activation of rhodopsin and we have this into metarhodopsin. We saw that CGMP is reduced by the end of the day if light is there. But if darkness is going to be there, everything's going to be opposite. Rhodopsin will not be activated. Okay, you will have there will not be reduction of CGMP. There's going to be CGMP there because nothing of this happens. So CGMP is not converted into GMP. So you'll have CGMP. In fact, you'll have more CGMP there. Okay. So in darkness, there's going to be more of CGMP. So what CGMP is going to be this night watchman? This night watchman is going to open up the sodium channels. So now what happens in dark? All the escaped sodium are going to go back from the inner segment to the outer segment. Therefore, what do you think happens now? Here is negative outside, but all the positive has gone back into the rod. So there is no polarity there. There is no poles opposite, nothing is there because whatever has came out has got inside. So there is no polarity is existing. So it's called as depolarization. So remember D and D. In dark, rod is depolarized. Dark, rod, depolarized. Then depolarization happens. Now, I said only in rod hyperpolarization, you have this, we have this peculiar mechanism. But in fact, there's going to be a small current is going to happen. Even though it's depolarized, it's depolarized. You will have some current. It's called a dark current or it's called a standing potential. Just remember that. But the key feature is that when you have depolarization of rods, here glutamate is going to get released. There we had rod getting hyperpolarized. So glutamate was not released in light. Here there's going to be depolarization of the rod. So glutamate is going to get released. So what happens when glutamate is getting released? Like I said, the BPC will be hyperpolarized, just ulta, just opposite of daytime. Here BPC is hyperpolarized. When there's hyperpolarization, like I said, first physiology, there will not be conduction of electrical impulses. So nothing happens in dark. Despite the fact that rod is depolarized, you don't see electric impulses being conducted. Clear? So this you should understand the differences between the dark and the light. So to summarize both, what happens in the daytime when light happens? There is no CGMP because night watchman is not there in light. There's going to be light is going to hyperpolarize the rod. No release of glutamate. So BPC becomes depolarized excitation. What happens in night? The CGMP, the night watchman will open up the gate. So it's going to be dark. It's going to depolarize the rod. As a result, it's going to be release of glutamate. And this, your BPC, bipolar, is going to remain hyperpolarized. So this is going to be a concept which you have to reproduce in your exams. So whatever diagrams I've drawn, I've tried to kind of split the diagram so that it's easy for you to follow, but you can put everything together like this in your exams. Okay, now you might wonder why I'm saying all these things. Like I mentioned always, please connect your basics to this. Connect your physiology always to a clinical application. In your exam, if you're going to put this table, the examiner will go crazy because here you have brought important genetic conditions, retinal diseases, retinal dystrophies into this picture by applying the knowledge which you have learned from this. Remember rhodopsin? Mutation of rhodopsin, there are hundreds of mutations of rhodopsin. Rhodopsin mutation will cause retinitis pigmentosa, especially autosomal dominant retinitis pigmentosa. Remember transducin? The rod transducin is going to cause congenital stationary night blindness. The phosphodiesterase, CGMP, mutation of these two things can cause autosomal recessive retinitis pigmentosa. Remember guanylate cyclase, the GC, which is going to secrete CGMP. When GC is mutated, it is often seen in Leber congenital amaurosis or LCA, which is a variant of or a very severe type of RP, an autosomal recessive type of retinitis pigmentosa. is going to be something which we'll discuss in a minute. This is going to be a transporter called rod ABC. It's called as ATP binding cassette. ATP binding cassette. Okay, ATP binding cassette. So ABC transporter, which is encoded by this gene called ABCA4 gene, which very often you will come across in Stargardt's disease. When you read about Stargardt's, this ABCA gene mutation is implicated. So what this ABCA gene does is going to produce this protein, this transporter, this rod ABC transporter. When this is not working properly, you'll have Stargardt's. So put this in your exam. It makes much more useful. And now you know why we are reading all these things. Now you will understand the relevance why we're reading all this physiology. Okay, now coming back, we said that when light strikes, this 11-cis retinal is going to become all trans retinal. Now we know that the two reactions which we thought, which is studied some time back. So it's going to be isomerization to all trans, rhodopsin is going to become activated. This is going to have this phototransduction. Now, this is where we come to the next topic, this Wall's visual cycle. What happens with this all trans retinal? Now, when keeps on becoming all trans, at one point, you need to regenerate this 11-cis, right? You need to once again bring back, because we keep on seeing things, we need always regeneration of this 11-cis retinal. How it happens? So again, when light strikes, I said this 11-cis retinal becomes this 11-cis becomes the all trans, it's not 11's, all trans retinal. So this all trans retinal is going to be accumulated. This all trans now has to go out. Once light is hit, once phototransduction has happened, all trans gets accumulated. Now all trans should come out. How it comes out? We'll see from now on how this all trans is going to get regenerated back to 11-cis. That's Wall's visual cycle. Now, this is the outer segment of the photoreceptor. This is going to be this disc, which is going to have your all trans retinal, like you saw in this picture. Now, this all trans retinal is going to come out. It is going to bind to this ABC transporter protein. You remember this ABC transporter protein we just mentioned for Stargardt's disease? So this ABC transporter protein is going to be like this auto rickshaw, which carries your all trans retinal. So this rickshaw man is going to take this guy, all trans retinal, into the cytosol of your photoreceptor, outer segment. This is the outer segment in toto. Okay. This is going to be just the lamellar disc. This is going to be cytoplasm. So in the cytoplasm, this ATR is all trans retinal is going to be dropped. With the help of retinol dehydrogenase enzyme, this all trans retinal becomes an alcohol form called as all trans retinol oil. Now, this is going to go out into this interphotoreceptor matrix. In other words, in the interstitial space, interphotoreceptor matrix is going to come out. In this interphotoreceptor matrix, this all trans retinol is going to take another auto rickshaw. This is not an auto rickshaw. It's going to be like a big cab. It's like a bus. It's going to combine with this interphotoreceptor retinol binding protein. Interphotoreceptor retinol binding protein. Now, this auto rickshaw, so this bigger cab is going to carry this guy back to RPE. So what enters RPE is going to be this all trans retinol is going to enter the RPE. So in RPE, it's going to come again with another retinol binding protein. Even if you forget this retinol binding protein, it's okay. But don't forget these steps. So this RBP, you have another RBP inside. It's called a cellular retinol binding protein. So this all trans retinol is going to bind with CRBP. Now it's the time when the all trans retinol becomes isomerized into 11-cis retinol. 11-cis retinol. Okay. And this 11-cis retinol is going to become 11-cis retinal. Finally, 11-cis retinal is going to be formed with the help of retinal dehydrogenase. So this is an important enzyme. This is like an isomerase called isomerohydrolase RPE65, another important enzyme which is implicated in genetic diseases. Just know this. So the retinol becomes retinal with the help of retinal dehydrogenase. Over. So now we have regenerated the 11-cis retinal. Now 11-cis retinal is regenerated. Our job doesn't end. Now again the cycle continues, right? So again this 11th retinal has to come out, go back into your disc membranes. So 11th retinal is going to go back, combine with to form the rhodopsin, activate rhodopsin. So the cycle continues. So I'm going to bring back the summary slide what I discussed first. So vitamin A is going to go to the RPE, retinal becomes Leven's retinal. This Leven's retinal is going to enter the outer segment of rods, combined with rhodopsin, and there is going to, rhodopsin is activated to form the phototransduction. Okay. The Leven's retinal is going to convert it into all-trans retinal, all-trans retinol, taken back to the RPE, remember, here to Leven's retinol. So this Leven's retinol is going to get regenerated back. This is the Wall's visual cycle or retinine cycle. Some called retinine cycle, Wall's visual cycle, just visual cycle. So far, I think I've made things very clear. Now, we are 1 hour and 5 minutes till now. I have few slides on the retinal layers and a brief function of them. Shall I continue or can we stop it here? That's going to be my question.
No, sir. Please continue, sir.
Good. Okay. So there's no harm in exceeding the time.
No, sir, not at all.
Okay, good. So the basic cells forming retinal layers. So just give me a minute. So this is just to complete the proceedings. These are, you all know that the retinal layers, like 10 retinal layers, and you have only very few cells forming. One of the commonly asked Viva question is that, tell me those 10 layers of retina, okay, which most often we try to memorize without actually knowing what is forming or what are forming those 10 layers. But knowing those 10 layers is crucial in understanding the anatomy of the OCT as well. Now, RPE, retinal pigmentary epithelium. The rest is going to be called as a neurosensory retina that you all know. Now, you have 4 types of cells here. It's called as neurons, interneurons, glial cells, and vascular cells. These 4 cells are going to form the neurosensory retina. Now, what are the neurons? The neurons are nothing but your photoreceptors, your rods and cones, your bipolar cells, your ganglion cells. These 3 things going to form the neurons. On the contrary, the interneurons are going to be 2 important cells called as the horizontal cell and the amacrine cell. The glial cells are going to be 3 cells, MAM, microglia, Muller cell, and astrocyte. We'll all discuss in a minute. Vascular cells are going to be 2 types, endothelial cells, and it's going to be your pericytes. Pericytes and endothelial cells. So please have these things in mind. Now, what are the functions of your RPE? Now, we just discussed a very important function of RPE. RPE is going to help in the regeneration of the vitamin A. It's going to be playing a crucial role in your vitamin A, in your Wall's visual cycle. And not just that, it's going to absorb the scattered light. So light is going to get absorbed to the RPE, and from the RPE, it's going to get transferred to the rods and cones. And more importantly, it forms the outer blood retinal barrier thanks to its tight junctions. The visual pigment, like I said, the formation of vitamin A is very important with the RPE. Synthesis of growth factors. One of the crucial functions of RPE is the phagocytosis and digestion of the photoreceptor way. So all those rods which are like dead and decomposed will be taken up, will be phagocytosed by your retinal pigmentary epithelium, the outer segments of your rods. Horizontal cells. Now, this recent update. Previously, they were telling that horizontal cells are going to be these neurons which are connecting between rod and bipolar cell. This is what would be given in your standard textbooks. But what I am teaching you is from the latest edition of AAO, from American Academy of Ophthalmic. So recently they say that horizontal cells are going to be these antagonistic neurons between cones, between 2 cones, not between bipolar cell rod cells, between 2 cones. Please remember that. It's again quite a complex mechanism how this horizontal cells play, but just understand horizontal cells and amacrine cells, both cells are going to be inhibitory. They will try to keep things under control. Remember, horizontal cells are going to be inhibitory mechanisms to the rods, which means when you have light hitting on the rod or cone, to be more specific on the cones, we do not overloading of cones because light will keep on hitting us, kind of bleaches off, right? So we need an inhibitory mechanism also to make sure that you have a proper contrast. And also when you come from your dark to light and light to dark, To adapt those things, you need to have these feedback mechanisms, the inhibitory feedback mechanisms. That's what horizontal cell does for the cone. So when you have this cone cell hyperpolarization, what happens in light? The horizontal cell is going to inhibit further impulses from going on. Okay, and horizontal cell is going to have this GABA, which is going to be neurotransmitter. In the contrary, glutamate is going to be the neurotransmitter from the rods and cones. So GABA is going to be the inhibitory neurotransmitter from the horizontal cells. Amacrine cells are nothing but cells which are going to be between the bipolar cell and between the ganglion cell. The same concept. Amacrine cell is going to have this feedback input. A negative feedback is going to be given so that it is going to limit or is going to filter out unnecessary information or unnecessary impulses. Muller cell is going to be probably the largest or the longest cell available. It's called as a backbone of retina, which extends from your ILM. Okay. So it's going to span almost the entire retina. Please understand, some books give the term as interplexiform neurons. Now we don't have this term existing. So there is no more this interplexiform neurons. There's going to be only two important interneurons. That's going to be your amacrine cells. It's going to be your horizontal cells. This interplexiform neurons no more considered. This Muller cell is a part of your glial cells. Your Muller cells, your astrocytes, and microglia. All these things are going to be an important component of these glial cells. So these are very important in cases of maintaining retinal integrity, okay, in providing nourishment, and also making sure that the structure is going to be properly maintained. All right. I think I have finished for what I wanted to convey. Now, again, it's quite difficult to cover the entire physiology within 2 hours. So what I've done is I've tried to focus more on those topics which are difficult to read by yourselves with some recent updates. And I hope I've given you enough information which you can handle or kind of take things forward. My references, what I would say, would be AAO. So in AAO, in your second part, the part which discusses the principles of ophthalmology. So there you have like 100 pages of physiology. So please read that. Once you read those 100 pages, you will be like very confident and you will have a conceptual overview of the entire physiology in a nutshell. And then you can read Qurana, then you can read whatever books you're usually reading so that you can add more points to that. So read AAO first if you're going to be a first-year resident. Then take the journey into Qurana, because Qurana can be quite an overwhelming book. It's very comprehensive, quite exhaustive book. 50% of topics what are dealt there may not be asked in your exams. So you need to pick and choose from Qurana. You can't read cover to cover. That's where AAO comes into play. AAO gives you a better overview. You can clinically correlate concepts better, and then you can switch to Qurana if you need. I hope I have given the references and also kind of given you a guide on how to read and how to answer. I think the slides, whatever I've shown you till now, would have given you an idea of how to go about. Please reproduce whatever I've put on my slides, because the slides are nothing but hand-drawn or handwritten notes. So there is no reason for you not to copy and put it. It's quite straightforward. That's why I've not intentionally used any textbook images, but rather I've stuck to my own illustrations just to show that you can easily reproduce those images or those illustrations in your exam papers also for getting better marks and for easy understanding. So if there are any questions, I would be happy to answer.
Yes, sir, we have had quite a lot of questions, but you have covered it so beautifully well that your lecture encompasses all these questions and they are very well answered. The students can just have a look at both the videos prior to their examination, and I'm very sure that these diagrams, the line diagrams, are going to be very reproducible in the exams, so that they find it easy to express it to the examiner, that they know the concept, and at the same time save their time upon writing lengthy questions and answers for the same. Before we call it a day, I have a small announcement to make. We meet next on the 19th of July, which is Essentials of Ophthalmic Pharmacology, Part 1 by Dr. Tirumurthy, sir.
Yeah,
Thank you so very much, sir, from the bottom of our heart, for spending all your time from your busy clinic towards all the postgraduate students. I'm sure they'll really benefit from both these lectures when viewed together right before the examinations.
Yeah, and I really loved that you didn't stick to the old school PowerPoint presentation and made physiology-like topics so interesting. So thank you so much for that.
So much, Shafari. Thank you so much. Once again, thanks to Santosh sir for inviting me as a faculty for this. I feel very honored and humbled. In fact, iFocus is living my dream. iFocus is doing something which nobody else is doing. They're giving best possible content, bringing experts all around the world, and to give you the best possible learning experience, which is my mission and my goal eventually, to give the best teaching experience for my students. So I'm very happy and honored to be a part of this. Thanks a lot. Thanks a lot for that wonderful team. Thank you so much.